Assalamualaikum dan salam sejahtera kepada semua. Selamat datang ke program E-mobiliti UPM-IPB 2022 yang bertemakan Divided by the Sea, Connected Virtually. Pada pagi ini, para peserta akan menyertai kelas 2 session yang akan disampaikan oleh Dr. Muhammad Nur Hisham dari Jabatan Biologi, Fakulti Sains Universiti Putra Malaysia. Sebelum memulakan kuliah kita pada pagi ini, saya menjemput saudara Luqman Hakim untuk membacakan doa bagi memberkati majlis kita pada hari ini. Dipersilakan.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله الذي بنعمته تتم الصلاحات والصلاة والسلام على رسولنا الأمين وعلى آله وصحبه أجمعين. Ya Allah, kami merafakkan rasa kesyukuran di atas keizinanmu untuk kami melaksanakan sesi Dynamic Fusion bagi program E-mobiliti anjuran Persatuan Mahasiswa Fakulti Sains Universiti Putra Malaysia dan Himpunan Mahasiswa Biologi Institut Pertanian Bogor. Kami juga bersyukur kerana Engkau mengizinkan kami menghadirkan diri, emosi dan roh kami sehingga dapat menyelesaikan program ini dengan jayanya. Ya Allah, bagimu segala puji dan puja. Kurniakanlah kami lisan yang lembut basah, mengingati dan menyebut namamu, hati yang penuh segar mensyukuri nikmatmu, serta badan yang ringan menyempurnakan ketaatan kepada perintahmu. Ya Munzilar Rahmat wa Ya Fatihal Barakat, kembangkanlah lembayung dan gelombang rahmatmu kepada kami, ibu bapa kami, pensyarah-pensyarah kami, serta sahabat-sahabat seperjuangan kami. Ya Allah, Ya Qadi Al-Hajat, ampunkanlah dosa-dosa kami, didiklah hati kami untuk mengutamakan keperluan saudara-saudara seagama kami, sebagaimana kekasihmu mendidik para sahabat baginda berbuat sedemikian. Satukanlah hati-hati kami dan berikanlah kami ruang dan masa untuk bantu-membantu dalam menegakkan amal ma'ruf wa nahi munkar. Allahumma ja'al jam'ana haza jam'an marhuma wa tafarruqna min ba'dihi tafarruqan ma'suma wa la taj'al lahuma fina wa la ma'na wa la man yataba'na syaqiyan wa la matrudan wa la mahruman. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azaban nar wa sallallahu ala sayyidina Muhammadin wa ala alihi wa sahbihi wa baraka wa sallam walhamdulillahi rabbil alamin.
Amin ya Rabbal Alamin. Terima kasih saudara Luqman atas bacaan doa sebentar tadi. Sebelum saya menyerahkan majlis kepada doktor, izinkan saya untuk memperkenalkan latar belakang doktor. Nama diberi Dr. Muhammad Nur Hisham Muhammad Nazif dan kepakaran beliau adalah dalam bidang fisiologi dan pengeluaran haiwan, pemakanan, endokrinologi dan bioteknologi reproduktif. Kini dijemput Dr. Hisham untuk meneruskan pembelajaran kita pada hari ini. Silakan Doktor.
Hai, Assalamualaikum. Selamat pagi semua.
Selamat pagi Doktor.
Can I hear respond from IPB?
Waalaikumsalam.
Salam. Okay, before we actually start, boleh tak you semua on cam untuk saya kenal wajah-wajah baru daripada keluarga seberang sana? Rizky saya dah nampak, Zarina, Eva, yang lain tak buka lagi ni? How many IPB students that we have here? Rustia?
Around 32, Doktor.
32?
Yep.
Okay, boleh tak buka cam? Nanti while I'm giving a sharing session, you may off your cam. I just want to see your faces. Handsome and beautiful faces. Ni pagi-pagi ni harapnya semua ceria-ceria, sudah sarapan. Because you're gonna hear a lot from me today. So I hope you won't get bored lah ya. So are you okay if I mix languages? Malaysian, Indonesian, English. Okay ya? Bisa ya?
Yeah, we are okay, Doktor.
It's okay.
Okay, saya ramai family di Indonesia, especially in Lombok, di Bandung, di Jakarta. So I can speak a bit of Indonesian. Sebab kita tengok sinetron banyak kan, so I can speak a bit. Alright, so dengan saya, you don't have to be shy. If you have anything to ask, just stop me. If you want to share your thoughts, you are most welcome to do so. Alright, so Bismillahirrahmanirrahim. Assalamualaikum again. My name is Dr. Hisham. Tadi siapa tadi? Najihah. Already introduced myself a bit. So basically, I'm also an alumni from Department of Biology, where some of the biology student here and also Faculty of Science student here. I'm also an alumni in 2002. So dah lahir ke belum? I did my degree in 1999. I graduated in 2002. So semuanya ni rata-ratanya umur berapa ni?
20.
20, still young. I am half of your age. Okay, so hari ni dah tahu ke apa topik yang kita akan dengar bersama? You can unmute your mic ya. Restu. Sorry,
yeah.
Zarina? Yes?
Assisted reproductive technology in farm animals, Dokter.
Okay, assisted reproductive technology. Farm animal, of course you know lah, farm animal kan haiwan ternakan di ladang. But what is assisted reproductive technology? Kalau diterjemahkan dalam bahasa Malaysia ataupun bahasa Indonesia, apa itu?
Mungkin reproduksi yang terbantu ya dokter. Jadi istilahnya itu kita mencoba agar hewan tersebut itu bisa mempunyai keturunan mungkin sederhananya itu.
Okey, keturunan yang baik-baik kan, betul. So kenapa
Benar,
kita perlu bantu haiwan-haiwan ini tidak bisa mating sendiri, tidak bisa reproduce sendiri? Why we as a researcher or human being need to help them in reproduction? What do you think so? First two.
Yes.
Nah,
Menurut saya agar meningkatkan keberhasilan dalam mating, jadi tingkat reproduksinya lebih tinggi lagi.
okay, maksudnya kemungkinan some of the animal not really in terms of reproduction not really good. Okay, bakanya itu mungkin tidak bagus. Jadi kita perlukan baka yang baik daripada another animal from the same breed or species. Okay, that is one of the reason. Another thing is tadi Rizky bilang to get a superior breed. Okay, we wanted to produce a superior breed by having a crossbreeding from different breed but in the same species. Okay, we cannot breed from different species but in the same genus we can just to crossbreed to have and to produce a better quality and superior gene of animal. Okay, all right. Okay, should I share my screen now? You need to allow me to share, host. You need to click on multiple participant can share kan?
Ya, dokter.
Dah? Okay.
Dah.
Can you see my slide now?
Yes.
Right. Yeah, I cannot see the slide. Okay, all right. All right. Bismillahirrahmanirrahim. Assalamualaikum. Okay, so we shall start now. So our topic today is assisted reproductive technology, or we call it ART, in farm animals. So my name is Dr. Muhammad Hisham Muhammad Nazir. You can call me Dr. Hisham. I'm from Department of Biology, Faculty of Science, UPM. So this is my address and details. If you wanted to contact me in future, please do so. Okay, this is a bit about me. So my research is actually on animal physiology in broad, but I'm looking at the nutrition aspect, the hormone, endocrinology is hormone, and also reproductive. That is my research interest lah. So if you can see here, there are so many animals that I show in the slide. So these are my my favorites. Okay, my favorites species to look at to do research. Okay, I did my master in goat, as you can see up there. Oops, sorry. Up here is the goat, and I also work with cattle or cow when I did my PhD. And then I'm currently working with one of the research institute in Malaysia, which is Mardi. We are working on village chicken, and specifically product by Mardi, which called Ayam Mardi, as you can see in the picture. And then also I have a project with apa itu perhilitan di Malaysia, iaitu badan yang menjaga hidupan liar di Malaysia. So I also have research project working with Sambadia, rusa unicolor. And you can see here in the middle is the nutrition part lah. So I work with many types of greens or fodder that can be the food source for the animal, and I see how the animal actually react towards the nutrition feeding regime. Okay, so I'm also a lecturer in animal physiology. So on top of the picture there ada gambar apa itu cow fetus. So that is one of the topic that I teach in my course. All right. So assisted reproductive technology, as you mentioned tadi, dia adalah teknologi pembiakan bantuan ataupun berbantu. So why we need to help this animal in reproduction? Kita akan go through one by one what are the reason after this. So ART divided into two types, field-based, iaitu di ladang, and di makmal laboratory. So there are two types of ART being conducted. So what are the ART conducted in field-based? This is some of the ART that being conducted, such as estrus signalization and supervision. And then we have artificial insemination. And then we have embryo transfer. Okay, so may I ask if you know what is estrus? What is estrus? Restu, are you talking? You need to unmute. I couldn't hear your voice.
Estrus itu seperti siklus ovulasi tapi terjadi tanpa melalui peluruhan rahim.
Again, please. Estrus is what?
Estrus itu seperti menstruasi tapi terjadi pada hewan selain seperti tidak seperti manusia. Jadi rahimnya itu tidak melurus setahu saya.
Okay. Betul ke Zarina? Did Restu tell us the truth?
Yep. I agree with Restu. So.
Okay. So estrus is actually kita tak panggil menstrual ya. Menstrual tu only in human. So kitar haid itu only in human, but in animal they don't have kitar haid. They don't have menstrual cycle, but they have estrus cycle. What it means by estrus is that the animal is having a reproductive event where the dominant follicle bursts out and release the matured oocyte where ovulation occur. So in common language, we say the animal is on heat. Kalau bahasa Melayunya is berahi. Okay, berahi bermaksud the female is ready for copulation, for mating with the male. Okay, so reproductive-nya ialah aktif. Okay, so dia release oocyte and then if copulation occur, ejaculation occur, so the sperm will swim to the oocyte and fertilize, and that's what we call estrus. Alright. How about super ovulation? You dah dengar ovulation. Ovulation is apa tadi? What is ovulation?
Release egg cell from ovary.
Okay. Yeah, where dalam di ovary tu ada so many follicles. Inside the follicles we have oocyte or cell telur, okay, eggs, and then if the dominant follicle mature enough, they will burst, and their burst tu akan release the oocyte. So that one we call ovulation. But super ovulation means we wanted to have or produce more follicles to burst out and release more eggs at one time. That's why we call super. All right. And then we have artificial insemination, which is permanian beradas, where we do the fertilization outside of the body, or we use a tools or device instead of having two animal, male and female, having a mating. We as a human help them to insert the sperm into the female, and then we have another embryo transfer similar to the artificial insemination, but instead of using sperm, but we are transferring embryo. So yang dah fertilized, dah menjadi embryo, dah apa tu, dah menjadi dah dari live organism lah. So we transplant into a recipient. Alright. So today I'm only be focusing on the field base. There are so many other ART being conducted in laboratory. So as you can see here, we have IVMC, we have microinsemination, nuclear transfer, sexing gamete, embryo, and gene transfer. All this must be conducted in the lab where it involves drugs, medium, other equipment that very specific and delicate work to do in the laboratory before they can transfer to the animal. All right. But the field base, it can be very straightforward. And sometimes it also involves some part of the procedure need to involve in the field, and then the rest of the procedure was conducted in the field. All right. Okay. So let's recap a bit what is reproduction. So there are two types of reproduction, as you know. I'm supposed you should know this by now. There are asexual reproduction and sexual reproduction. So today we are only focused on the sexual reproduction, where it's the creation of individuals from the fusion of gametes, which is sperm from the male and eggs or oocyte from the female, through fertilization to form a zygote. So it involves meiotic division, and of course it has higher genetic variation compared to asexual reproduction. So I don't have to explain the asexual reproduction because it's a very basic knowledge, and I assume you all have already learned about the asexual reproduction. Okay, so for sexual reproduction, it must involve the gamete from the male, which is again sperm, and then gamete from the female, which is ovum or oocyte. So both undergo the internal fertilization to produce zygote, which containing polar body, which indicate that that is an active zygote or a live zygote. All right, but in order to fertilization occur, the main thing, because as you know, the ovum dia bergerak ke? Did oocyte move around in the reproductive tract of the female?
Enggak, dok.
Enggak.
Dia tinggal di ini, di tuba fallopi. Untuk
Akan
spermanya ke atas.
Ya, di fallopian tube atau di bahagian apa itu? Uterus body. Alright, so it won't move. So apakah gamete yang akan move around? Apa gamete yang akan move around?
Sperm.
Siapa yang jawab tu?
Sperm.
Betul, sperm. Okay, but what are the characteristics? What are the important parameter for the sperm in order the fertilization to be successful?
Menurut saya hal-hal yang berpengaruh tuh kayak kesehatan sperm itu sendiri dan tingkat keasaman gitu kan, dokter, kalau nggak salah.
Kalau kesihatan sperm itu contohnya apa?
Jadi saya pernah dengar tapi ini di manusia sih dokter. Jadi kayak
Apa?
kalau nggak salah tuh kalau kesehatannya tuh kayak termasuk kayak bentuk fisiknya, terus kayak kekentalannya kayak gitu sih dokter yang saya tahu.
Ya, betul. So sama aja. In human or in animal, the sperm must be able, first thing is must be able to swim. How the structure of the ovum is like a tadpole, kan? But different species might have different structure. Probably the head might be different, but they all have tails, which help the sperm to swim around. And what is the aim of the sperm? What is the aim of the sperm? Apakah matlamat utama sperm itu? Come on guys, you should know this. Apa itu?
To fertilize the egg.
Yes, to reach to the oocyte first, kan? So kalau semua ada billions of sperm, the aim is one, okay, to fertilize with the egg, kan? So dalam banyak-banyak, imagine the oocyte is you, kan? Diva, the only diva there. So there are so many fans wanted to meet you, but you will only accept one or two. So the most important thing is that the mortality of the sperm is high, the mortality is low, and the mortality is high. Okay, what is mortality? Ini orang yang sama je jawab ni. Mana IPB yang lain ni? IPB? Anandito. Can you unmute?
Yes, sir.
Okay. What is mortality? Sperm mortality, what does it mean?
Sperm ability to move.
Okay. How about mortality? Tadi ada dua ya, mortality and mortality. If you say mortality is movement, well, how about mortality?
Can you spell it, sir?
Mortality, M-O-R-T-A-L-I-T-Y. Another one is M-O-T-I-L-T, mortal, M-O-T-I-L-I-T-Y. Mortal. One with R, one with R.
Mortality is the sperm death rate, I guess.
Baru betul. Tadi you sebut mortality itu movement. Mortality is movement. Mortality is the rate of death. Okay, kematian sperm itu. So good sperm is they move actively, which is high mortality, and low mortality, kurang kadar kematian. Alright? Okay, banyak soal ya, takut ya? Jangan takut ya. Saya memang macam ni. Tanya Izwan. Because I was known as biomaleficent in bio. Okay. So, what can you observe from this video? Did you see any movement? Did you see any movement tadi?
Yes, sir. I can see any movement.
Banyak ke sikit?
Yes, sir.
Biasa-biasa je kan?
Ya.
Ini kita katakan if the movement you nampak macam alah, slownya, perlahannya, itu bermaksud dia low mortality. Dan bila low mortality, you tengok berapa banyak sperm yang you nampak bergerak. Ada yang tak bergerak pun kan, compared to this one. You nampak ni semua ni? So kat sini, dia actively move. Actively move means dia high mortality and low mortality, which is kurang yang mati. Sebab if you can see, banyak yang bergerak. So ini menunjukkan this is a good quality sperm. So in order fertilization to occur, we need this kind of good quality sperm. So they're able to move fast and very less sperm that has died lah. Okay. All right. So question, how many motile sperm are required during insemination to achieve pregnancy?
Satu je.
Satu.
One.
Yang lain? Only one? Ada jawapan lain tak? Tak ada? Okay, so minimum. Kita ini untuk insemination ya. Tadi tu yang fertile. Yang soalan bertanya, ni tricky question dia. Dia tanya how many motile sperm are required during insemination to achieve pregnancy. Dia tak tanya how many motile sperm are required for fertilization. Kalau fertilization, yes, only one. But for insemination, sama ada insemination means kita bantulah with assisted reproductive technology tadi, kita gunakan artificial insemination. So per insemination, we need at least 6.7 million sperm, motile sperm per insemination. Sekali kita inject sperm dalam female, we need at least 6.7 million sperm motile and high mortality and low mortality. So kita nak banyak yang hidup dan banyak yang actively move. So how is actually we wanted to observe this? So kita ada sperm evaluation before we do the insemination. Sebab kita nak buat insemination, of course we wanted to use very good quality sperm. So we cannot simply ambil sperm daripada mana-mana male without looking at the quality, kan? And then terus inseminate. So we might inseminate sperm yang low quality, which lesser than 6.7 million per insemination. Okay, faham eh?
Oh, dah.
Orang UPM yang jawab. So, yang ini baru betul. How many sperm are required to fertilize a mature egg? Barulah jawapannya only one. Okay? So, only one sahaja yang diperlukan untuk fertilize unless they have 2 eggs and then they need 2 sperm because it will produce kembar tidak seiras. Kalau misalnya from one sperm, from one oocyte and then they split, that one will be identical twins. Okay, got it? Now, the development of embryo. Okay, so we have from zygote to embryos lah. So once the egg being fertilized with the sperm, so it becomes zygote. And then zygote will divide it into 2 cells, 4 cells, 8 cells, and then it become morula. Then from morula, it become compacted morula. And then blastocyst and the haplocyst will implanted into the body of uterus of the female. Ada soalan ke tu? Itu jawapan. Oh, okay, jawapan. Okay. Host tolong bacakan ya. I won't notice sometimes. Okay, so now let's recap ya. Reproductive system. So for male, there are ovary. Hormone involved are estrogen. So estrogen functionnya ialah for female sex characterization. So in male we have testis. So the hormone involved is testosterone. So the function is to trigger or to help in characterization of male sex. Okay, so in female the estrogen tu ada 2, progesterone and estradiol, and they also have a bit of testosterone. Okay. Okay, this is a female cow. So this is the diagram only. I will show later on the real reproductive tract of the female. So this is cow. Cow is actually how we call female cattle. Yeah? So cattle, kalau female, betinanya kita panggil cow. Okay. And then this is the reproductive tract of the cow. Okay. So nanti kita akan go through one by one its structure, what are the names and what are the function of its structure. And then we have male, we call bull. Yeah? Bull is only for male in cattle. Okay. So this is how it looks like for the reproductive tract of the male. All right. So bull reproductive tract. So here, these two structure ni, this is the testicle or testis. Yeah. So it's the site of sperm production and testosterone synthesis, and I think perhaps by now you should know sperm contain half of genetic makeup and oocyte contain half of genetic makeup. Yeah, so XY kan. And then we have scrotum is the structure of the skin that protect the testis and regulates the temperature. And then we have epidermis, where the sperm was stored and maturation development occurred in the epidermis. And then we have vas deferens to transport sperm from epidermis to urethra. And then urethra is a tube inside of the penis to transport sperm and urine. So dia ada dua function lah. One is for urination, one is for transporting the sperm. Okay. And then we have penis, which is the main and crucial part for copulation, for mating. Yeah. All right. So basically, these are the function. So the seminal vesicle is to protect the sperm, fluid containing fluid to protect the sperm. And then we have prostate gland. So dalam male, human or animal, male pun ada. So di situ akan produce milky fluid to nourish the sperm. Okay. And then we have corpus glands, fluids that cleans urine out of the penis. So dia ada function untuk dua function ni berbeza ya. So bila nak urinate, so corpus glands will play the roles. When to ejaculate the sperm, so prostate gland involved to nourish the sperm. And then we have sigmoid flexure, where it is a muscle extend penis out of the body. So pernah lihat tak structure bull, reproductive tract of the bull in real life? Have you seen it before?
Pernah.
No, tu dah pegang semua tu.
No, I haven't seen it before.
Yang biology dah pernah lah kan, dalam my class, siapa yang dalam my class maybe dah pernah tengok. So the function the penis tu bukan sentiasa di luar badan. So that's why dia ada sigmoid flexure ni, where is a muscle dia akan bila untuk melakukan copulation, barulah bila the penis is erect, this muscle functionnya ialah untuk mengeluarkan penis out of the body. So that bila penis itu keluar, so dia akan penetrate the vagina of the female, and then berlakulah copulation and mating. Bila ejaculation, lepas ejaculation barulah this structure pula, the next structure, retractor muscle, dia akan pull back the penis back into the body. So penis for most of male in animal only go out of the body bila masa untuk melakukan mating. Okay, bukan sentiasa di luar. That's why they have two muscle ni lah, sigmoid flexure and retractor muscle. Satu untuk membantu mengeluarkan keluar daripada badan untuk proses copulation, dan selepas selesai mating proses, retractor muscle akan pull back into the body. Okay? Clear ya? Ada soalan setakat ini?
No, sir. Clear.
Clear Okay. Now we have heat or prepuce. Ini adalah macam external skin yang memprotect the penis lah. So sebab penis tu dia tak ada protection kan. So bila dia di luar, So dia ada sheet lah. So bila sheet tu macam kulit sajalah, skin. Okay, to protect the penis from injury. Alright. So kita lihat sedikit apakah hormon yang terlibat dalam bulls. There are GnRH, gonadotropin releasing hormone. Okay. So it's control the pituitary gland, and then we have LH, luteinizing hormone to produce testosterone. We have follicle stimulating hormone, hormone needed to produce sperm, and then we have testosterone, other hormone to produce sperm and cause body change and develop libido. So what is libido? Tahu tak apa itu libido?
Hasrat seksual mungkin Pak.
Again?
Hasrat seksual. Untuk
Sexual desire. Yeah.
Ya,
Sexual
desire.
desires.
Libido is keinginan seksual. Okay, so testosterone important. Kalau low testosterone, so dia tak akan ada libido. So the male won't have interest untuk melakukan copulation or mating. Okay, so if the testosterone is low, this is not a good quality bull lah. So we won't use this bull for any insemination. Okay, so this is not a good bull lah. Maksudnya dia hormon tu tak cukup, so dia tak akan produce or ada keinginan untuk melakukan mating. Alright. So next is I cannot see lah. Okay, this is how the hormone works lah. So hormon tak lari banyak lah, mesti akan involve dalam anything dalam to regulate our body kan, mechanism. So gonadotropin releasing hormone will stimulate the pituitary gland to release LH. When the LH increase, it will stimulate the intestinal cell for pembahagian sel, the growth, proliferation of dendritic cells. And then when LH is decreased, so androgen like testosterone will increase. This will stimulate lah the development of reproductive part, and then the secondary sex characteristic, and also the sex drive, which is the libido tadi lah. Okay. Okay, how do we actually, okay, kita tadi nak macam I keep on mentioning about the quality of sperm kan. So as researcher, how we actually do the assessment, and before kita nak buat assessment tu, of course we have to collect the sample. So in this case, we need to collect the semen kan, for us to determine whether this bull has good quality sperm or vice versa. So by that, there are two types of procedure how to do the how to collect the semen from the bull. One, we use a device which called electro-ejaculator, and the other one we use artificial vagina. So electro-ejaculator ni, there is a device which dia akan ada sedikit aliran elektrik. Not too much, just enough to trigger and stimulate the bull to ejaculate. So dia akan insert the device which is called electro-ejaculator through the anus, because as you can see here in the picture, the prostate gland ni nearby dengan dia punya saluran anus. Ini saluran najis dia. So when you insert the device here, so it eventually will trigger or stimulate the prostate gland, and then dia akan stimulate the bull to ejaculate. And then kita akan collect lah semen tu, and we observe under microscope to determine whether it's a good quality sperm or poor quality sperm based on the characteristic, morphology of the sperm, whether it's normal structure or abnormal structure. And then we look at the movement, whether it's high mortality or low mortality. And then we look at whether the kadar kematian itu tadi rendah ataupun tinggi. Okay. So I stop sharing kejap. So I tunjuk satu, mana ni? Okay, video. Can you see this video?
Boleh, Doktor?
Yes, sir.
The one that I'm showing now.
Feet,
is
prep use to ensure that he
apa ni
should be physically capable of the
menggunakan
stamina required to get his
electro
weight of females
ejaculator.
in
So
calf.
first dia akan examine lah the
Secondly, a
bull
reproductive
kualiti ke
examination
tidak, dia akan measure
of his
the
testes, checking for the
testis
tone and feeling for
size,
lumps,
and then dia
then
rasa ada
measuring his
lump ke
scrotal
tidak,
size.
whether it's
A kick
normal
or
or
rump bar is
abnormal
needed for
before they
this, although
actually
very
choose
few bulls
the
object
bull.
to it.
So as you can see, I
As
just mentioned they
testicles are
measure
basically his semen
the
factory, so
diameter
size is important.
of the testis, whether
Small
it's normal
testicles have
or
been associated
abnormal.
with bulls having
Kalau
exceptionally
maybe
good
it's
muscling.
too big
But what
or
use
it's
is this
too
if he cannot
small,
pass it on to
maybe
his
dia
offspring
indicate
because he's not very
some health
fertile,
issues
or
lah.
passes on fertility
Okay,
problems to
lepas
the
kita
offspring he does
examine
produce?
everything,
We also
dia
examine
ni
his
sih
accessory
saya
sex
kata
glands
tadi dia
internally.
akan masukkan the
His
device
penis is
electro
usually seen
ejaculator
while
tadi.
stimulating him to give
Nampak
his
tak?
semen sample.
Nampak tak the penis start
Thirdly,
erected?
the
This
semen
is the electro
examination.
ejaculator.
This
It's
is
very
usually
huge
collected using the
because it's for
electroejaculator, which is a
big
battery-powered
cow,
probe which
big
goes into the
bull.
rectum
So
and
they insert
stimulates the nerves to the
through
accessory
the
sex glands of
anus,
the penis.
and then dia akan switch on
Unfortunately, these nerves are
the
right
device,
next to some
so
of the
dia
nerves
akan
in the
ada
back
sedikit
legs.
macam
So the
vibration.
legs will
So
straighten each time the
electric
machine is on.
stimulation, dia adalah certain reading dia, and then dia ada dah tube tu dia dekat bahagian penis untuk collect the semen. It's very quick, so
Then we
dia
look at
tak
the
ada
semen
menyebabkan stress pada animal.
grossly to check it is
And
not
quickly,
contaminated by
this is
urine
the
and assess its
sperm
consistency,
assessment lah.
which
So they
roughly
will look
indicates the
under
concentration of
microscope
the sperm.
to determine whether it's a good
Next, we look
or
down the
poor
microscope, looking
quality
at overall
sperm.
wave motion.
Okay, so this
Although
is
this is interesting to
electro
look at,
ejaculator.
this does not.
All right. Now, the other one is using artificial vagina, kan? So it's a simple device where kita akan menggunakan female yang on heat or on estrus tadi to stimulate the guy, the bull ni, untuk having standing mounting, memanjat bahagian belakang betina in aim so that the male tu nak seolah nak penetrate the vagina lah, but then we have that artificial vagina to collect the sperm. Okay, you see how it
तक जरूर देखें क्योंकि आज की वीडियो हमारी नस्ल सुधार के ऊपर है और नस्ल सुधार में हमारा सबसे ज्यादा जरूरी होती है एआई और एआई के लिए हमें जरूरत होती है एक अच्छे सीमन की और उसी सीमन के बारे में आज आपको डिटेल में बताऊंगा कि कैसे उसको कलेक्शन की जाती है और कैसे आप बेहतरीन किसी अच्छे सीमन बैंक का सीमन यूज करें.
So this is the artificial vagina, so they collect semen. And that's it. So female tu untuk buat menggunakan artificial vagina, the female must be on estrus or on heat, berahi. Dia actively sexually dia active, maksudnya ada ovulation occur lah. So dia maksudnya dia animal ni dia ada senses lah, dia ada hormon yang boleh detect the animal tu on heat ataupun tidak, kan, because from the urine they can smell, from the mucus discharge from the vagina, the bull can know that this female is ready for mating. Okay, got it?
Yes, Doctor.
Okay, I back to my notes. Okay, so clear ya? Ada soalan ke tu? Saya nampak dia chat ni. Pernah Izwan. Okay.
Sir, saya ingin bertanya mengenai metode elektroejakulasi itu. Nah, ada alasan nggak kenapa kita harus kayak sedikit memberikan rangsangan kepada prostat gland agar female-nya itu ejakulasi. Mungkin kenapa alasannya itu ejakulasinya itu di prostat gland bukan di penisnya. Seperti itu, Pak. Mungkin pertanyaan.
Okay. So, as I mentioned earlier, structure animal ni bukan macam human. The penis is outside the body. But for bull, the penis is inside the body. Kan tadi? Jadi, kalau kita tak boleh nak merangsang dia daripada luar, so that one will cause injury lah kalau misalnya animal tu rasa stress because you touch the private part of the animal. So dia akan rasa macam dia akan rasa seperti mahu di attack because suddenly you touch the part the sensitive part of the body. But using the electro ejaculator device, So dia akan kita akan dapat lakukan proses ini very quick because the rangsangan should be at the prostate gland. The prostate gland, because kat situ tadi, if you go back to the previous lecture, previous note, tadi, so prostate gland tadi is the milky fluid to nourish the sperm. So kat sini nanti, bila sperm daripada testis tadi itu keluar ke sini nanti, so yang prostate gland ni harus able to produce the milky fluid tadi untuk nourish the sperm. So bila dia rangsang, so prostate gland ni akan make sure that the sperm is healthy lah, sebab dia tu diberi milky fluid tadi kan, so that dia boleh swim ke dalam, keluar daripada body melalui penis. So rangsangan tadi tu, bila dia rangsang di bahagian prostate gland, so ejaculation tu akan berlaku dengan cepat compared to where you touch the penis, and sometimes penis tu tidak keluar because rangsangan tu tidak berlaku di prostate kan, and then so the sigmoid flexure muscle ni tidak akan membantu the penis go out of the body. Okay, make sense?
Yes, dokter. Jadi berarti di prostat tersebut itu banyak saraf terasa ya dibandingkan di bagian kepala penis seperti itu.
Yes, prostate gland itu penting sebab dia kat situ juga yang merangsang libido tadi.
I see.
Okay.
Thanks dokter. Ya, paham.
Okay, so macam tadi pun kalau guna artificial insemination pun kita tidak merangsang bahagian penisnya kan, tapi kita menggunakan female yang reproduktifnya aktif which on heat. So the male know, they can sense the female that on heat. So dia sendiri dah terangsang di situ. So maka dia ejaculate. Bila the moment the bull start to mount the female, memanjat the female, so libido, the trigger, the stimulation already occurred. So that's why they ejaculate. Okay? But compared to ejaculator tadi, there's no female around the bull. But then dia menggunakan electro-ejaculators to stimulate the ejaculation. Alright? Any more questions? Ada lagi soalan? Tak ada? Ada ke soalan? Tak ada dah?
Tak ada dah.
Okay. Now we proceed with cow reproductive tract, which is the female. So this is how it looks like. This is real specimen. So we have ovary. We have two sides, the right side and also the left side. And then the function of the ovary is to produce egg, and egg containing half of the genetic makeup. And then we have uterine horns, which is for filter-bearing animals to develop. So kalau misalnya berlaku fertilization, so the hatch blastocyst will attach to uterine horn and untuk development of the fetus lah. So bila dia membesar, dia akan proceed to the uterus or body of uterus. So di situ body of uterus or uterus is the site of embryonic development during gestation period. So sepanjang tempoh pregnancy itu dia akan membesar di bahagian uterus. Okay, it started from uterine horns. And then bila fetus itu membesar, dia akan turun ke bahagian uterus dan membesar sehingga saat parturition ataupun saat kelahiran. And then we have cervix, which connect the uterus and vagina and serve as barrier during pregnancy. So bila tiba masa, kalau misalnya tak ada cervix ni, kalau sentiasa ada bukaan nanti tak cukup bulan, so dah berlaku abortion. So cervix is important because it's a bukaan ataupun laluan for parturition ataupun kelahiran. Bila fetus tu dah membesar dan cukup bulan, tiba masanya cervix akan dilate ataupun buka untuk proses kelahiran berlaku. Alright? And then we have vagina, which is the female organ of copulation and mating, and also the opening of bladder. And then we have infundibulum, which to catch eggs after burst from the ovary. So dekat sini, this area, lepas follicle to burst, oocyte akan keluar. So infundibulum ini akan catch the eggs, okay, before they transport ke uterus. So oviduct ataupun in human we call it fallopian tube is a site of fertilization and transport of embryo to uterus. Sekiranya berlaku copulation and fertilization occur, so dia akan develop to fetus lah, embryo. Jika tidak, so dia akan keluar melalui kita head in human, in animal pun akan keluar melalui mucus discharge. And then we have vulva ni di luar, kita akan nampak dengan mata kasar di bahagian luar badan female, which is the external opening untuk proses copulation tadilah from the male and female, okay? So this is the site of ovulation. Gambar ni saya ambil daripada group IZWAN for my course comparative reproductive biology. So di sini adalah ovary from the female cow. So this is the site of ovulation. So you nampak ya, nampak macam dah pecah kan? So jadi parut, scar. This is what we call ovulation. This is ovulation site. Okay. So after the ovulation occur, so site of follicle tadi yang dah pecah tadi menjadi parut tu, we call it corpus luteum. So corpus luteum ni adalah scar of follicle yang dah pecah tadi, yang ovulation site tadi. So inside of the corpus luteum is something like this. So dia ada function dia lah. So bila kebiasaannya bila ovulation occur, corpus luteum ni akan maintain progesterone level to be in high level concentration sebab nak maintainkan pregnancy. Sekiranya fertilization occur lah. Okay? Am I talk too fast ataupun okay?
Okay, doktor.
Okay. Now, this is comparison lah between ovary yang tidak ada ovulation dengan ovary yang ada ovulation. So, as I mentioned earlier, female ada dua ovary, left, right and left. So normally one side will having ovulation, the other side takkan ada ovulation unless twins yang tidak seiras lah, maksudnya kedua-duanya akan ada ovulation. So they will need two sperm, ada two eggs. So fertilization occur, then they will produce two calf lah, dua anak, two fetus. Okay. Okay. Tadi ini rasanya dah tunjuk tadi kan, what ini female reproductive untuk haiwan apa?
Kelembuan.
Apa?
Cow.
Cow. Okay, this one? Teka-teka.
Kambing.
Kambing, this one?
Rusa.
This one?
Kuda.
Kuda. Okay, this is cow, correct. This is horse. Salah ya, ini this is not horse. So this is horse. This one is goat or sheep. And finally this is sow, female pig. So kalau you sekali tengok, the structure is almost the same. Mungkin yang bezanya mungkin dekat bahagian uterus ni lah. Ada sikit beza compared to yang sow, female pig. Okay, dia a bit macam not really bentuk seperti tanduk. Okay, but for cow, horse and goat, they have similar structure, almost the same. Okay. Alright, so female cows, they also have gonadotropin releasing hormone, which control pituitary gland, triggers LH and FSH hormone production. So FSH is important for follicle development, which eventually will produce the eggs or oocyte. And then we have estrogen produced by the follicle to start the heat period. So estrogen ni dia dah nak due to ovulation tadi, okay? And then when LH increase, so rupture of eggs from the ovary means ovulation occur, and then progesterone will blocks FSH and LH production, and then prostaglandin akan menghentikan progesterone production bila pregnancy occur. So progesterone ni akan keep high untuk maintain the pregnancy bila sekiranya pregnancy berlaku. Sekiranya pregnancy tidak berlaku ataupun abortion berlaku, so progesterone will drop lah, akan jatuh to the low level of concentration. Okay, so yang ni I takkan go through in detail. So puberty. Puberty ini ialah in human pun we call puberty, iaitu tempoh matang, sexually active, so and become functional. So puberty adalah umur di mana reproductive organs of the female or male tu become functional. So dia boleh melalui proses ovulation and whatnot. Alright. So in cow, normally it occur pada umur dia 6 bulan sehingga 12 bulan, 6 months to 1 year. So that is normally the age of female cow become active lah, the reproductive organ become active, means dia dah ready untuk pregnancy and whatnot. And then female reproduction occurs within estrous cycle. So estrous cycle adalah reproductive cycle occurring when pregnancy doesn't occur. So estrus tadi saya cakap heat or receptivity to mate. So dia matang dan dia on heat ataupun berahi. So sekiranya tidak ada pregnancy berlaku, bermaksud estrus cycle itu akan berulang. The same event, the same cycle will continue until pregnancy berlaku. Bila pregnancy berlaku, dia dah tak ada estrus. Dia stop for cow. Gestation period is about 9 months like human. So sekiranya lembu betina itu pregnant, so estrus cycle tidak akan berlaku, dia stop. Okay. Untuk cow, we call it as polyestrus. Maksudnya dia cycle every year except when they got pregnant. Clear ya? And then once dia on estrus, so cycle itu biasanya akan berlangsung sepanjang tempoh 21 hari in average. So the range between 18 to 24 days. Ini macam human lah. Kita haid kan, kita menstrual cycle tu dia ada tempoh-tempoh dia. So same goes with animal. But kita tak manggil menstrual cycle, but we call estrus cycle. And then fertilization is where egg and sperm unite. And then gestation adalah tempoh pregnancy. Parturition is act of giving birth. So gestation period for cow, just yang I mentioned tadi, is 9 months, about 280 days to 285 days. And then parturition is act of pregnancy. So this is just fun fact about age of puberty in each animals, especially in farm animals. So for cow, 6 to 12 months. For sheep, 5 to 7 months. For sow, 4 to 8 months. Horse, for 12 to 15 months. And human, 9 to 16 years. So it's different cycle, yeah, because that why kita panggil menstrual cycle different with animal. Okay, the menstrual cycle normally we call for cow ialah polyestrus, which is cycle throughout the year. Sheep, seasonal, normally decreasing light. Peak sexual activity was in fall, which is in September to November. And then so polyestrus like cow, and for horse, seasonal, but dia perlukan daylight yang panjang. So sexually activity during spring, which is in March to May. The length of estrus for cow lasts for 19 to 23 days. For sheep, 14 to 19 days. So about 19 to 23 days. Horse, a bit longer, 10 to 37 days. So duration of the cycle once dia berlaku, it will last 12 to 18 hours for cow. For sheep, 24 to 38 hours. Maksudnya once dia on ovulation, dia akan last about 24 to 36 hours. For sow, 48 to 72 hours. For horse, it's about 3 to 8 days. So dia longer, so dia lain sikit. And then from estrus, once dia detect dia on estrus, before ovulation berlaku, the burst of the egg tu, dia ambil masa sehingga 24 to 32 hours. Because estrus ni dia ada early estrus, middle estrus and late estrus. So bila dia dah late estrus tu, maksudnya dia dah towards to ovulation. So this is the duration lah before they actually ovulate. Okay, for sheep, 24 to 30 hours. So 36 to 42 hours. And for horse, 5 days. It lasts for 5 days. So within these 5 days, kalau dia nak buat insemination ataupun nak buat natural mating, this is the time lah within 5 days after ovulation. Okay. So number oocyte for cow about 1 egg, sheep pun 1 to 3 eggs. So so about 10 to 20 eggs, and horse only 1 egg. Okay. Now, we look at the field based study lah yang kita nak tengok hari ni ialah estrus synchronization and superpopulation, artificial insemination and embryo transfer. So, estrus synchronization tadi you tahu ialah haiwan tu akan on heat kan. So, estrus synchronization ini ialah kita nak coordinate a batch of female. For example, a batch of 20 goats female to have the same time of estrus. Maksudnya katakan hari ni 10 hari bulan. So kita nak 20 hari bulan 3, all 20 female goats tadi tu will become on heat on the same day, same time. Okay, that what we call estrus synchronization. We synchronize the estrus. Whereas the superovulation means kita induce the production of many mature ova or oocyte from the ovary, and kita akan menggunakan synthetic drugs to help or to superovulate the ovary so that they can produce more mature oocyte. Okay. Biasanya kita involve superovulation ni because kita nak buat ART lah. Alright. Ada soalan setakat ini? Tak ada. Clear ke? Am I talking too much or too fast?
Tengok okey je tu. Tak lagi
Okay,
sangat.
Okey.
Tak mengantuk ya? Dah sarapan ke belum ni?
Tak tu.
Dah. Okay, you may have your drinks or you can eat, but you boleh apa ni lah, switch off your cam lah. Nanti kan kawan-kawan you lapar, you triggered, you stimulate their hunger. Okay? Alright. So, when I said drugs use or involve drugs for superovulation and synchronization ni tadi, there are few types of hormones, synthetic hormones that we need to use lah to stimulate or to mimic the same hormone that they have in their body. But because we wanted to help them produce more and better and good quality punya reproduction punya event, so we need a synthetic drugs lah, which is safe for the animal and also for the human lah. Because for example, progestin, it prevents estrus and extend the estrus cycle. So tadi kan I dah tunjuk setiap animal tu dia ada estrus cycle yang berbeza. Jadi bila kita nak synchronize, so setiap animal ni macam manusia juga. Ada orang datang menstrual cycle hari ni, menstrual kita haidnya hari ni. Your friend might be next week. So kita nak untuk animal ni sebab kita nak dia pregnant at all the same time. Biasanya kita akan buat estrus synchronization ni because you wanted to have them pregnant at the same time and deliver or have parturition at the same time as well. So progestin ni one of the example yang in commercial punya drugs is control intravaginal drug release device, or we call it SIDA. This one I use for my master research. So it's nanti I tunjukkan gambar dia. So this progestin ni dia mengandungi synthetic progesterone. So when we insert into the vagina of the female, for example goat, kan? So dia akan releasekan hormon sintetik tadi tu, the progesterone yang sintetik tadi tu, slowly into the bloodstream. So what happen bila progesterone tu daripada SIDA ni masuk dalam bloodstream, so the level of progesterone will increase. Kan? Bila increase, bermaksudnya apa? Estrus ni, bila nak estrus, progesterone mesti low. Okay, kalau misalnya dia tinggi, dia tidak akan datang berakhir. They will never go through estrus. Okay, tak akan berlaku estrus because the level of hormone tinggi, specifically progesterone. So that is how it work lah, how we actually manipulate the hormone. And then prostaglandin ni dia akan membuatkan female itu on heat ataupun dia akan memendekkan kita cycle-nya. Biasanya bila ini involve dalam superovulation lah, so dia akan inject hormone through the muscle injection. So dia akan masuk dalam bloodstream. So dia akan increasekan hormon prostaglandin ni untuk menyebabkan haiwan itu on estrus, on heat. So maknanya progesterone tadi akan kurang. So prostaglandin akan tinggi untuk menstimulate proses estrus itu berlaku. Okay, so generation juga dia responsible lah untuk release follicle hormone which is FSH and luteinizing hormone LH from the pituitary gland untuk membantu or stimulate ataupun trigger ovulation berlaku. So dia akan menyebabkan ada follicular wave dan banyak folikel-folikel yang akan jadi matang untuk yang mengandungi mature oocyte, dan kemudiannya bila ovulation berlaku akan banyaklah mature oocyte jadi, because this one kita nak tadi super ovulation jadi kan. Alright. So this is example of estrus synchronization and super ovulation regime lah untuk cattle. Yang ini kita buat untuk artificial insemination. So kita synchronizekan estrusnya dan kita super ovulate supaya dia produce more mature oocyte. And then bila kita dah dapat ada ovulation berlaku, we use artificial insemination protocol where we insert good quality sperm from probably imported bull yang kita nak, yang ada good history, ada good gene, superior breed lah, and then we inject into the reproductive tract of the female. So this is how it works. This is how the procedure lah. Kita dia ada regime dia, day one what you need to inject, and then kita masukkan sida ni untuk control the estrus. Once we remove the sida, we need to inject hormone prostaglandin ni untuk trigger ovulation. So within 72 to 84 hours, this is where we need to observe estrus itu berlaku ke tidak. Jika berlaku, then baru kita lakukan artificial insemination, plus we inject more hormone to help the successful of the artificial insemination. Alright. So this is the sida I mentioned tadi. So it looks like Y shape. So why, the reason why in Y shape, because once you insert into the vagina like this, like this, dia mula-mula macam kapsul. Bila dia masukkan dalam vagina, so dia akan terbuka. The reason why dia terbuka macam ni supaya sida itu tidak tertanggal daripada vagina, takkan jatuh. Because here ada string di bahagian bawah ujung ni, bila kita dah cukup tempoh, katakan 7 hari di dalam vagina female tadi, kita akan remove. Dan baru kita masukkanlah hormon-hormon yang ada kat sini tadi yang kita nak ni, PG. This one vary lah, dia try and error dalam research which one produce more good results lah. Okay. So and then any injection of hormone, and then yang agak tadi through intramuscular, and then they will go to the bloodstream. Alright. Tadi I mention what is estrus kan. So how we wanted to detect the animal is on estrus ataupun tidak. So the first one is the swelling of vulva, and then ada mucus discharge, and then the behavior tu kita akan nampak dia keruan, resah, dia bergerak sana bergerak sini, and then nampak and then dia akan letih, dia akan banyak berehat pula lepas tu. And then dia akan menunjukkan libido kepada male, di mana dia bersedia menunjukkan minat kepada male untuk melakukan proses mating tadi. But the specific sign yang confirm that female is on estrus, where we observe visual active mounting, which shown in the picture. So if you observe the male memanjat the betina, and then you see the penetration occur, itu bermaksud confirm that female is on estrus, meaning there are ovulation occur, sebab ada sel telur yang keluar daripada ovari dan menunggu sperm untuk fertilization. Clear eh? So ini saya tunjuk video. This is what I meant by female receptivity towards male. Dalam kandang ni adalah male, this one adalah female. Ni nampak tak, this female dia twiggling tail? Nampak tak?
Nampak tak?
Nampak tak?
So this indicate that this female also on estrus. Okay, tapi kalau kita observe betul-betul dekat bahagian vulva dia, there might be a mucus discharge. So nampak tak this male semua dah macam tak tentu arah ni, semua menunjukkan minat towards this female. Nampak? This indicate that this female is on estrus. So jantan yang this female accept, ni kita akan tengok the next video. So this is standing mounting. This process we call standing mounting. jantan ini memanjat betina while the betina keep on twiddling her tail. If we observe carefully, di situ mungkin nampak swollen vulva, warna pinkish, and then ada mucus discharge. Nampak tak jantan tu duk smell the vagina area because dia ada release hormone. Okay, so tadi yang ni, this is the standing mounting. Bila nampak macam ni dan berlaku penetration, so confirm lah copulation tu berlaku. Alright? So, kalau bila animal tu on estrus, so what we do next is artificial insemination, sama ada mating tu berlaku sama ada natural mating ataupun artificial insemination seperti dalam gambar rajah ini. This is natural mating and this side on the right side is artificial insemination. Okay? So, this is my project during my master's punya study. So, this is what I did. My project is actually memang on estrus synchronization and superovulation in goats. Okay, saya lihat which regime the best for goat. So as you can see on the right side ni ialah saya withdrawn blood sample from the vena jugular to see the hormone profile untuk indicate at what day actually this goat become on heat. Okay. So this is the hormone profile lah. So I actually take the blood on daily basis every day. So for estrus synchronization, I ada different types of duration. Example kat sini I bagi 17 hari. So I ada buat 10 days, 14 days, 17 days and 21 days. So that is my treatments. Okay. So this is how it look like lah dalam hormon. Kalau you tengok kat sini. Yang pink one is the progesterone, the blue one is the estradiol. So these two hormone very important lah dalam female. These are the two hormones that indicate whether the female is on estrus or pregnant, sebab dia sentiasa agonistic, dia sentiasa opposite dia punya hormon. But kalau misalnya tak ada apa-apa reported event berlaku, so dia akan macam ni lah, akan remain high or remain low. So selepas 17 hari, I remove the sida and then akan ada buat super ovulation using the drugs. Kat sini saya guna apa tadi tu? PGR. And then lepas saya cabut, once I remove the sida, 2 hari selepas itu, dalam tempoh 24 hours to 48 hours, the female should be on estrus. How I detect dia on estrus? Because visually kita observe tadilah twiddling tail, nampak standing mounting and what not. But for hormone profile, nampak tak bacaan ni? I highlightkan dengan warna merah ni. So kalau you tengok level dia ni, I tunjuk. Dia bawah 1. Progesterone ni bawah 1 nanogram per mil. So dia bawah the threshold. If the bawah 1 nanogram per mil, it's confirmed that animal is on estrus, which indicate ovulation berlaku. Okay, so this is the ovary. So this is dominant follicle. So bila estrus bermaksud kat sini estradiol akan meningkat, progesterone akan menurun. So bila estradiol yang warna biru ni tinggi, nampak dia opposite progesterone rendah, estradiol tinggi. So estradiol ni akan tinggi pada estrus, indicate that there are more growing follicles were developed. As you can see here, ini dominant follicle, ini dominant follicle. So banyak follicle yang matang di bahagian ovary ni because estradiol dia very high. So bila dia ovulation berlaku, so progesterone ni should be basal level which is below 1 nanogram per mil, which indicate kat sini one of the follicle might burst. Okay, might burst, ovulation occur. So itu how the estradiol and progesterone dia punya relationship is inverse relationship. Okay, dia akan jadi negative relationship where satu tinggi, satu rendah. Okay, boleh follow?
Ya,
boleh follow? Ke nak rest 1 minute, 2 minutes? Or should I proceed?
You can proceed, sir.
Sorry?
We can follow. So just
Okay, alright. Let's proceed.
Yeah.
So once tadi dah buat oocyte synchronization, then I dah buat superovulation, then it's time for me to recover the oocyte. Kita dah do all the manipulation kan, so we wanted to collect the oocyte for next research lah. So in this case, this is me, masa ni I very slim lagi this was in 2006 when I did my masters in Universiti Malaya. So this animal, lepas kita dah buat proses estrosynchronization and superovulation, this animal will kita akan bawa ke makmal untuk kita recover the oocyte untuk embryologists do further research lah, whether to do embryo transfer and what not. So this is me, kita akan use this animal, the female ni akan pengsan lah, and we make sure that the female akan dipuasakan, tak boleh diberi makan dan minum untuk mengelakkan injuries throughout the surgery. So animal ni dah diberi anesthetic, so dia akan dibius, so dia akan unconscious. So lepas tu kita akan taruhlah anesthetic di bahagian yang kita nak tebuk. So basically it's not open surgery, it's just a matter of puncturing at few parts, a very small incision, just to make sure that we have camera to view the parts of the reproductive tract, and then we have aspiration to flush and suck the oocyte that we wanted to retrieve, and then we have macam penyepit untuk hold the ovary so that it will move lah while we are actually sucking or flushing the oocyte. I will show you the video. This is how we call it excellent superovulation, because there are many matured dominant follicle. Ni sebab superovulation, the function is to produce many matured oocyte kan. So many matured oocyte is from matured or dominant follicle like this. So kalau tengok bengkak-bengkak ni is like macam jerawat pimples. So bila dia dah masak, it's about time to burst. So this is how it look like for ovary. As you can see here, this one device which is hold the ovary so that it won't move. Then we have another needle where we punctured the dominant follicle to flush and suck the oocyte masuk dalam tube. Okay. And then the embryologist will indicate whether the oocyte is a good quality oocyte or poor quality oocyte. So we have a grading system, as you can see here, grade A, B, C, and D. So how we indicate that the oocyte is grade A when they have 5 layers of cumulus cell. This is the oocyte, then they have cumulus cell around the oocyte, and the layer is more than 5 layers. That is grade A, and then it become lesser to indicate poor oocyte. Kalau grade D ni bermaksud dia naked oocyte, dia tak ada cumulus cell. Means it's very poor, and the chances of the egg to being fertilized sama ada artificially ataupun naturally, chances of dia being fertilized is very low. So they're not good quality. Kalau sperm penetrate pun, it might not develop into embryo. So maybe development ataupun pembahagian sel 2 sel 4 sel tadi tidak berlaku. Okay? Faham ya? So this is how we indicate the quality of oocyte lah. Okay, let's watch the video. This is how we do the process. This is the procedure, the apparatus that being used. So small incision will be made on the area. This is my professor. So this one, that is camera when we can view this part. All right. And then this is how it looks like inside the reproductive tract. So one device is to hold, dia macam penyepit, and then this is a flushing needles. So it's actually puncturing the dominant follicle, and we recover the oocyte, and dia akan suck into this needle, and then dia akan masuk dalam special tube in special device, and then we will give to the embryologist to determine the quality of oocyte di sini. Okay. Alright. So now we go to another second ART, which is artificial insemination. So artificial insemination is actually a procedure by placing a semen, usually frozen lah, because biasanya kita import good quality sperm ni from negara-negara lain, atau kita nak special breed. So we import, or from the farm that we have, we kept that in in liquid nitrogen to froze them until we going to use that later in future. So it's normally the semen is obtained from superior male. This one kita dah tengok dah check dah dia punya quality. Tadi ada buat measurement dia punya testis, dia punya dah evaluate semen dia, high mortality, low mortality and high mortality. So bila semua tak ada abnormal semen, and then that one kita consider sebagai superior male lah, and we kept in liquid nitrogen until further use. And then AI ni ialah kita masukkan semen itu ke dalam reproductive tract of the female by natural mating, ataupun kita menggunakan device with the hope of causing a pregnancy. So this is artificial insemination using a device. This is the AI gun. And then the process is like this. I already mentioned to you. So semen collection, sperm analysis, once kita dah confirm the good quality, kita preserve. And then bila kita nak do the AI to active, reproductively active in female, dah choose, kita akan synchronize the estrus, and then kita akan involve drugs untuk super ovulate. And then the semen yang kita preserve tadi, kita akan thaw and kita akan inseminate using artificial insemination gun. Okay. Ini tadi ada cerita kita semen collection tu kita akan gunakan artificial vagina ataupun kita guna electro ejaculator. So this is the looks of the sperm morphology. So the normal should be oval shape lah for the head. Kalau dah kepala dia besar sangat atau terlalu kecil, ataupun dia bercabang, ekor bercabang, bentuk kepala ni tu abnormal, lepas tu bahagian lehernya bengkok. These are all the abnormal sperm. For cow, masih berupa bentuk yang seperti human. Tetapi for ayam, tikus, dia berbeza sikit. Kepalanya tidak bulat tapi runcing dan mungkin ada cangkuk. Okay. So semen analysis ni kita akan buat sperm count, number of sperm per number of sperm dalam sekali kita ambil tu, dalam satu titis tu ada berapa banyak sperm count tu, kita akan detect lah. This is considered sebagai mortality, kadar kematian dia low ataupun high. Mortality, percentage of sperm moving, and then sperm morphology. These are the three characteristics that we look at lah for semen analysis. So this is how we preserve the good quality semen. So this is the straw that we use. Then we kept in liquid nitrogen, which the suhu is minus 196 degree. Okay. So this is so many. Dalam ni banyak lah ni good quality sperm from superior breed, daripada male. And then this is again lah the details lah how we actually do the sterilization. And then when we wanted to inseminate, kita akan gunakan AI gun ni. It's a very simple device where we normally do this at the field. Okay, good semen insemination, sama ada kita gunakan trans-cervical AI, which kita akan gunakan masuk ikut vagina dia, or kita akan menggunakan the same procedure dalam video yang saya tunjuk tadi, but we do it at the field. Okay, so it can be done whether in the lab or in the field. So this is what we call laparoscopic AI. This is what we call trans-cervical AI. Okay. Okay, this is how it works. And then this again lah, ni how gambar yang lebih jelas. So dia akan masuk through the vagina. And then dia release lah. This is the gun containing the straw yang ada semen tadi. Then dia akan inject dalam reproductive tract of the female. So this is how we do it laparoscopic lah. So animal ni must be sanitized supaya dia diuse supaya dia tidak bergerak, because dia akan diikat kepada cradle. So this person, the expertise akan lakukan laparoscopic artificial insemination. Okay. Okay. This one is for large ruminant. So large ruminant kita tak boleh buat laparoscopic insemination, artificial insemination. So kita akan guna rectovaginal lah. So kat sini one hand akan masuk bahagian rectum untuk kita locate the cervix area, and then while another hand, katakan tangan kiri kita masuk ke rectum, tangan kanan kita pegang organ. Once tangan kiri dapat rasa the cervix area, so the gun should be insert through the vagina sampai ke bahagian permukaan permulaan atau pembukaan cervical tadi. Once kita confirm, baru kita inject the sperm slowly, macam dalam gambar bawah ni. This is the cervical, anterior cervical lah. Once the AI gun melepasi this area, baru kita releasekan the sperm. Alright, this is how it look like lah. Okay, dalam bentuk diagram, this is how it look like. So this method definitely need practice. You need to practice a lot. You need to get familiar where are the cervical area, so that once you dah tahu the structure, the location, then it will improve lah the successful rate of AI. Okay. So this is the advantage of doing AI. So they reduce cost. So kita boleh kurangkan tak perlu banyak animal di farm. So we just need the sperm or the semen, and then kita boleh overcome male infertility. So kita dah tahu dah which one is low sperm concentration, mana yang abnormal, kita exclude. So we only keep or use good quality sperm. And then breeding of animal across boundaries is very convenient because kita cuma transport frozen semen rather than we export or import the whole animal, which really very costly. And then kita boleh matekan animal ni dengan perbezaan size yang berbeza. Instead of natural mating, mungkin betina terlampau kecil, jantan terlampau besar, so they might cause injury during the process. And then by having assessment tadi, we can prevent lah certain disease daripada separate, daripada spread to all the animals in the farm. So we can control that. And then kita akan increase the genetic improvement because we only select semen from superior breed. Okay, and then the last one, continuous breed for superior breed ni using cryopreservation semen. So kita dah tahu dah apa yang kita nak, which breed yang kita nak, then this one is the advantages lah of AI. And then we also have artificial insemination in poultry, in ayam. So they have the same procedure. It's just that the device is smaller lah. So this is how it look like. Actually they use the small picagari je. So whether you can use the gun or you can actually use the small size of shrink. Okay. Okay, yes, this one I know. 20 minit, baru 10 setengah. Okay, so metodnya sama. So this is the process. So for female, for male ni, dia tak ada penis. Dia ada cloaca. But there's one structure looks like a tongue. We call it papilla. That is a structure that help deliver the semen. Okay, so this is how it look like. So very, very cloudy here. So this might probably indicates good quality sperm lah. Okay. So using shrink, we just insert the semen into the cloaca of the female, and we convert the bird lah after the artificial insemination by hoping the female will get fertilized egg. So this is how the hatchery in poultry. This is collection. So this process, definitely you need a practice. You need to massage the cloaca area at the back of the rooster so that they can ejaculate. So when papilla is protruded, so nampak tak tu semen tu ejaculate. So kita akan collect the semen using centrifugal tube or what, smaller tube. We collect and we do the semen analysis. Okay. So this dia punya differences in terms of color, ada yang rupanya
this is the chicken sperm
apa tu, watery, ada cloudy, ada ini watery, and then we have cloudy, and then creamy. So cloudy and creamy itu most probably from our observation it indicates the quality lah. So whether they're high or low, high mortality or low mortality. And then kita same procedure like we did in cow, we assess the sperm through the under microscope. So kita akan tengok lah. This is AI, how my student do AI. With small shrink. Okay, masukkan semen about 1.2 mil. And then we inseminate dekat bahagian kloaka female. We make sure the kloaka tu visible, nampak dengan mata kasar. See, nampak? Dan baru kita injectkan semen dalam kloaka betina. Okay? So how we actually determine whether the egg is fertile or infertile? So this student akan observe under simple method. Kita akan tengok, but we need dark room lah. So dia guna kotak saja, and we have lampu ni. So kalau fertile egg ni kita nampak dia gelap, and kalau kita see properly, kita akan observe ada macam vein webs, maksudnya ada kapilari darah. That one is fertile. Kalau dia infertile, so you nampak sini empty je, nothing, kosong, nampak tak, kosong. So that is infertile. Okay, spider like veins and dark spot for yang fertile. Infertile dia empty, tak ada apa-apa. Alright. So last one is ET. ET ni kita buat embryo transfer. So of course kita ambil daripada yang superior, yang the donor must be good quality, genetik dia bagus, sperm daripada jantan pun bagus, but the recipient tu tak perlu yang good quality sebab dia sebagai ibu tompang sahaja, because dia akan cuma digunakan untuk fetus itu develop dalam reproductive tract ni sahaja. Genetic comes from the superior bull that we select and good quality female yang kita ambil oocyte-nya, we fertilize them artificially, and then kita transfer embryo. Bila dia jadi develop menjadi embryo, kita transfer dalam recipient. Okay. So this is the bull selection tadi I cakap tadi, I dah explain. Recipient cows ni tak perlu, dia foster mother, so dia tak perlu good quality pun, as long dia healthy dan ada sejarah yang pernah melahirkan anak lah. Alright, so this is the procedure. Okay, this is second last part. I will go very quickly. So pregnancy or gestation is the process of where the fetus develop lah dalam bahagian peranakannya. So from this picture, can you spot the differences on left side and right side? What are the differences? Ada beza tak? Apa yang you nampak? IPB? Yang senyap sangat ni, mana ni? Ahmad Zahrani.
Yes, sir.
Yes. Okay. Can you spot the difference? Left side and right side. Apa difference-nya?
I can spot the difference.
You cannot spot any difference? How about the size?
The size of the intestine is bigger at the right.
My dear, this is not intestine. This is reproductive tract. This is reproductive tract. What is this? What is this? Ini kan uterine horn tadi kan? This is uterine horn. This is ovary. This is ovary. This is ovary. So what are the difference between the right side punya uterine horn and left side uterine horn? Apa yang you nampak? This is uterine horn. Uterine horn ni side of what? Siapa boleh bantu? Nesty, Laila. Nesty, Laila.
Ya, saya dokter.
Cakap kuat sedikit, kurang dengar.
Apa sudah terdengar?
Yes. So what are the difference? Can you spot the differences between these two?
The difference in the size of ovary. Jadi yang sebelah kanan is bigger than yang di sebelah kiri.
Okay, ovary ya.
Ya.
Okay, lagi. Irvana Sania. Irvana Sania. Ada tak? Terus hilang. Iqbal. Iqbal?
Yes, sir.
Okay. Tadi kawan you sebut ovary. How about you? It's very clear.
Is the tube of fallopian is on the right side is bigger than the left side, sir? The side of the vagina or the side of the ovulation?
Okay. This one, this is the uterus horn, uterine body, uterine horn, sorry, uterine horn. Nampak tak shape dia macam horn? This is the site where the fertilization occur tadi. So bila fertilization occur, dia akan jadi apa? Embryo kan? Embryo akan develop menjadi fetus. So inside here, if you can see the uterine horn on the right side is inflamed. Is bigger compared to the right side, the left side. Nampak tak?
Yes, sir.
Kan? So apa ada dalam ni? This ada fetus. So on the right side. So yang kiri ni, yang beza dia. This is very clear dia punya perbezaan. So left side is not pregnant, the right side pregnant. So this contain fetus. Fetus yang akan membesar for cow for 9 months. So this one probably the first trimester, which is probably 1 to 3 months. So dalam ni ada fetus ni. Okay? So pregnancy and gestation is the development of post-fertilization embryo into a fetus until the birth of animal or livestock. So as you can see here, so bila pregnant dia ada placenta lah. Okay, placenta ni a temporary organs that provide connection between embryo or fetus dengan mother dia. So dia akan dapat nutrition from the mother through the umbilical cord, and also there's this structure placenta, what we call in ruminant, also fungsinya ialah untuk memberi nutrition dan juga mengeluarkan bahan buangan daripada fetus. Okay, so this is structure of the pregnant animal in this picture showing cow lah. So as you can see here, this is the fetus. Okay, this is the umbilical cord. So this is the chorion membrane. Okay, yang ini adalah untuk bertindak sebagai uterine lining and help the fetal portion of the placenta. So dia sebagai apa, sarung lah, sarung amnion, kantung kepada baby tu. So dalam chorion membrane tu dia ada kita panggil amnion lah, amnion sac atau amnion membrane. Dalam ni ada amniotic fluid. So amniotic fluid ni bertindak sebagai buffer, sebagai untuk menyerapkan hentakan, sebab animal tu bergerak kan. So dalam tempoh fetus tu develop, so bila ada liquid ni, amniotic liquid ni, fluid ni, dia akan mengurangkan kesan hentakan tu dan melindungi fetus daripada any injury, because dia dalam organ dalaman ibu ada tulang-tulang kan, so dia akan protect the baby. Okay, so there are many types of placenta. So cotyledonary ini ialah biasanya pada ruminants. So this is what I show you just now. So this is Dia ada dua part. Placentum tu mengandungi karunkel, and then we have cotyledon. These two when they attached, we call it as placentum. So again, placentum ni fungsinya untuk menyalurkan makanan daripada ibu dan juga mengeluarkan waste macam carbon dioxide waste or bahan buangan daripada fetus through placentum melalui ibu. Okay. So as you can see here, dia attach bila kita tarik tu dia akan menjadi, one is caruncle and the other one is cotyledon. This both bila combine menjadi placentum. Okay, this is the placentum. So in cows, semakin tempoh pregnancy itu meningkat ke third trimester, the placentum will be bigger. So in cows, they have maybe berpuluh-puluh many placentums to help selain daripada menggunakan umbilical cord tali pusat. So diffuse ni biasanya pada equids, swine, and then we have zonal. This is normally pada carnivores such as dog, and then we have discoid normally pada human lah, and also pada rodent, mice. Kita ada placenta yang di luar. Okay. Alright. So ini binder sebagai permeability system untuk menjadi blood parent communication between the mother and also the baby. And then in early pregnancy, compositum ni pada ovari dia akan riliskan progesterone, so dia akan maintain pregnancy. So bila bila dah ke third trimester, so placenta yang akan ambil alih fungsi, the first trimester tu, the second trimester placenta akan ambil alih fungsi compositum untuk maintainkan progesterone concentration dalam blood system. Okay. Alright, ni I skip lah. So how we actually handle pregnant livestock? So kalau kita ada sebelum ni dia bercampur male and female, so we should separate the male from the pregnant females, and then kita akan take care of the pregnant ni, kita akan kurangkan menggunakan additive feed. So kita bagi yang natural feeding macam grass and everything. Vaccination, kalau misalnya ada birth compilation, kita akan menggunakan surgery with the help of veterinarian. And then milking cessation ni kita kena asingkan at least 2 months before giving birth. Asingkan kandang biar dia seorang-seorang untuk give birth and nanti akan menyusu anak. Okay. And then kita kena bagi balance feeding lah supaya both the baby, the fetus and the mother is healthy. And the balance feeding means the fetus will get enough nutrition to develop. Okay, so this is the fun fact, the size of the fetus during the first trimester. So this one is about 3 months. So you can see the size is about softball size. And for second trimester, this turn into bigger. So fetus mungkin akan rupa seperti tikus, sebesar tikus yang matang. So you can see here, sudah nampak dah. The structure dah clear dah, the ribs pun kita dah nampak, all the structure is very clear except for the eyelashes, the teeth and also the hair lah. And then bila third trimester ni pun sama, dah very clear, dah bone dah keras, the structure of the backbone pun dah dapat rasa, and then they might have eyelashes as well. So dia dah bony calf, so tulang-tulang semua dah rasa, dah keras, cuma gigi dia masih soft. Okay, parturition, last part. Parturition ni ialah saat kelahiran. So sama je human dengan animal, both are process of giving birth. So terminologies that we use in different animals. For cow kita panggil calving, process of parturition we call calving. For sheep, goat we call lambing. For sheep we call lambing, for goat we call kidding, and then for horse we call weanling, for cat we call queening, and for dog we call whelping. Okay, the terminology used for parturition in different animals. So an increased understanding of parturition process is helpful. Why? Because when we understand the behavior and then the physiology involved throughout the process, we can make very wise and sound decision whether we need to provide assistance or we don't have to provide any assistance. If the animal having difficulty to give birth, then definitely we need to assist them. Okay, then to assist them, we need to know lah the behavior and the physiology event of giving birth in animals. Okay, so what we need to do, we need to know the breeding date. Kita dah tahu dah dia dah berlaku mating, kita dah buat observation, we jot down the date when the mating is berlaku. Macam ni kan, kita biar natural mating biasanya kita akan letak macam pewarna pada jantan bila dia memanjat ataupun standing mounting, so we can see the mark on the female back. So this one indicates that copulation might occur dah, mating dah berlaku. So we jot down the date so we can predict lah bila this animal will give birth. Okay, the date is important lah. So this is okay, you tengok behavior ni, you beritahu apa yang berlaku. Okay, nampak ni apa berlaku ni? Teething tail and dash apa ni? What you can see here? Apa you nampak?
Mucus.
Ada mucus discharge and then dia restless, dia vocalize. So itu indicate apa? Nampak tak belly dia? Ni pregnant ke tak pregnant ni?
Pregnant.
So dia pregnant. So dia punya behavior sekarang dia restless, dia vocalize. And then dia twiggling tails, and you can see a mucus discharge from her vagina. So this indicate that this female is due to giving birth dah. So dah almost giving birth. This one, apa you nampak? Apa yang you nampak paling ketara kat sini? Did you see anything unusual?
Dia kaki tu je kejang tu.
Dia kejang. Ni apa ni?
Dia punya tits.
Ada dia dah swell, dah bengkak kan? Ada satu lagi. Can you see anything?
Something keluar daripada dia punya.
Ni, nampak tak ni?
Kaki dia keluar sikit.
Ni, apa ni?
Tu bahagian kaki anak dia kot.
Ni bahagian kaki anak dia. Izon pula yang jawabnya. Okay, so this is the nampak tak? Dah air ketuban dia dah pecah. The amniotic fluid tadi dah pecah, meaning dia ni dah nak keluar. So this female sedang melahirkan. So anak dia, kaki hadapan dia dah keluar. So ni tak lama lagi, dalam few hours, so animal ni akan beranak lah, melahirkan. So general signs of approaching parturition, so dia restless, nervousness, and then ada twirling, tail twitching, and then you can see the female is frequently urination, and then you akan nampak the other filling up, bengkak with milk, okay? And then we have engorged tits, so nampak bengkak tadi, and then the vaginal discharge, and then the belly drops, you dah nampak dah macam berat dah tu, dah memang dah hampir-hampir masa nak beranak, and then swollen, wet and reddened vulva. You can see amniotic sac observed ni bermaksud yang ni memang few hours lagi memang akan terus melahirkan. Alright. And then there are 3 stages. One is the preparatory stage where the cervical, yang I kata tadi, bahagian cervix tu akan buka. So bila buka, the baby akan keluar lah. Keluar tadi you nampak kaki akan keluar dulu. Posisinya macam ni. Okay. Macam tu. This is the cervical dilation, the opening. So the kaki yang utama tu akan keluar throughout the canal. This is the first stage. Then the second stage is the baby akan deliver lah. Okay, the fetus akan keluar melalui vagina like this. And then last stage is the expulsion of placenta. Selepas baby fetus dah keluar, few hours after that, the placenta will expel from the reproductive tract. So kalau misalnya tak keluar, itu membahayakan ibu, so we need assistance lah. Kita kena bantu melalui kaedah drug administration. Okay, so I tak go in detail. So this is how it look like. So the sign of discomfort, restlessness ini pada first stage. So dilation masa ni akan berlaku. So kita nampaklah pulse and respiratory rate akan increase, body temperature akan turun, and then the restless, kita nampak macam tadi kan, dia dah macam rasa sakit, dia nak beranak dah. Okay, so bila cervical tu keluar, nanti inilah first thing is the water bag. The first water bag akan keluar macam ni. And then the second water bag akan keluar mengandungi fetus. Kita akan nampak fetus tu keluar. There's some cases that the water bag tu tak pecah. But bila dah keluar nanti ibu tu akan pecahkan. Ada juga dalam proses masa the first water bag keluar dia dah pecah dan baby akan keluar. Biasanya when the water bag pecah je saja baby tu akan keluar dengan mudah. Okay. This is the stage of labor in cow. You can see here this is the first water bag. And then the second water bag, and then ni dah keluar lah ni, water bag dah pecah, so baby akan keluar dengan mudahnya lah. Okay, this is different animals, goat, sheep and whatnot. Okay, so duration ni berbeza lah. Second stage ni kau ambil masa 70 minit. Camel, beach, human ni ada different stage. Ada yang daripada beberapa minit sehingga beberapa hours Okay. Ni normal presentation. So keadaan anak itu perlu bahagian kakinya keluar dahulu. Sama ada kaki dan kepala keluar dulu ataupun dia songsang lah macam ni. Tapi kaki tu mesti keluar. Kalau dia tak keluar kaki, buntutnya dahulu, itu akan menyebabkan animal tu mengalami kesukaran untuk melahirkan. What we call dystocia lah. This is the position of the fetus yang akan menyukarkan kelahiran. Okay. Alright. I think that's all. Ni placenta ni, expulsion of placenta ni, what we call is after birth lah. So placenta tu mesti keluar. Kalau tak keluar nanti akan menyebabkan kematian pada ibu. So biasanya dia akan keluar lepas 30 ke 1 jam selepas the baby was born. Okay, dia akan keluar secara natural. Alright. Okay, I think that's all from me. Ada soalan tak? Ada apa-apa soalan?
Daripada saya tak ada ke? Mungkin daripada rakan-rakan daripada IPB mungkin ada soalan.
Have you seen whatever I have shown you tak? You dah pernah lihat ke? Or this is the first time you tengok?
For me, this is my first time to see.
How do you feel?
I don't know.
You don't know? Overwhelmed? Surprised?
Surprise.
It's good if you have opportunity to have hands-on experience lah. So like Izwan, Lokman, they have experience when they enroll my subject, comparative reproductive biology, where they have experience handling the animal by themselves. They can touch the ovary, they can actually dissect to look to the structure, what inside the structure, so they will learn better lah. Okay. Di chat box itu ada sedikit simple task for you to do. Ada 2 questions. It's just that I nak tahulah your feedback on what have you learned today from me. With that, thank you so much.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you so much, sir. Thank you for opportunity, sir.
Tolong jawab ya bahagian survey tu.
Okay, doctor.
Izwan, can you take a photo, Izwan?
Boleh tak cak Okay, first page 1, 2, 3. Saya senyum dulu 1, 2, 3. Okay, second page
Sekejap Izwan, tunggu dia orang buka kamera dulu.
Boleh. Oh, sorry.
Sini ada apa?
Kalau kod tentatif tak ada aktiviti tapi pelajar pada IPP ada kelas berapa.
Oh, okay.
Okay, semua ready dah ke? Ada lagi nak on camera? UPM, Alisa, Nat, Alia, Samiha.
IPB malu-malu ya? Why you are so shy?
Okay, ni tak nak buka kamera. Okay, saya teruskanlah. Okay, untuk first page. Ready? Okay, senyum 1, 2, 3. Okay, second page. 1, 2, 3. Okay, dah. Jadi saya akan share gambar.
Okay.
Terima kasih Doktor atas perkongsian yang sangat bermanfaat kepada semua mahasiswa pada hari ini. Dengan berakhirnya perkongsian sebentar tadi, maka berakhirlah kelas 2 session yang diadakan sepanjang 2 hari ini. Baiklah, sebelum itu, para pelajar Universiti Putra Malaysia diingatkan untuk mengisi link Google Form kehadiran yang akan diberikan di ruangan chat box dan komen di YouTube Live untuk kegunaan pemberian merit. Sekian, wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh dan jumpa lagi pada sesi Dynamic Fusion pada 12 Mac 2022.
Bye. Thank you. Take care, everyone. Stay safe.
Terima kasih, Doktor.
Thank you.
Bye, Doktor.
Thank you,
Thank you,
Dr. Noor.
Dokter. Thank you,
Thank you,
Dokter.
Doctor.
Thank you, Dokter, and thank you, UPM, for the lecture today.
Thank you.
Bye-bye. See you.